Okay, we're going to explain what osmosis is. Okay, so to understand osmosis, you have to understand what diffusion is. And of course, diffusion is the random movement of molecules from areas of high concentration to areas of low concentration. So hopefully you're okay with that. Now, with osmosis, you also need what is known as a semi-permeable membrane right there. Okay, so a semi-permeable membrane is a membrane that has got holes in it that let some things through and not other things through, and it will let water through essentially, but it won't let big molecules through. The cell membrane is in plant cells and animal cells is a semi-permeable membrane. Okay, so what we've got on the left-hand side of the membrane here, these are sugar molecules. For example, they're big. Now they're too big to get through these holes here. If you look at those holes there, they can't get through. If you look at, and then the blue molecules, these are supposed to be water, and they are small enough to get through the holes in the semi-permeable membrane. Now on the left-hand side, because we've got so much sugar, that's taking up a lot of the room. So there's not as much water on the left as there is on the right. So effectively, we've got a higher concentration of water on the right. And that means by diffusion, water is going to move in that direction. It's going to move across the semi-permeable membrane. And let's say this is inside the cell on the left-hand side. So it's going to move from the outside to the inside. All right. If you think about the sugar, well, the sugar is going to want to diffuse the other way, but it can't get through those holes. Yeah, the concentration gradient, would mean that the sugar would want to move from left to right, but it can't because it's too big to get through those holes. Now, in a cell, you have a lot of things dissolved in there. So inside a cell, you're going to have sugar and small molecules dissolved. And there's also bigger molecules like proteins, okay, and they take up a lot of space. So effectively, that's lowering the concentration of the water inside the cell. So if you put any cell into pure water, water is going to move into the cell, and that movement of water there is called osmosis. Okay, so you could imagine if we had a higher concentration of sugar I'll just say on the right hand side. So I'm going to just put loads of sugars in here. And what would they do? They would take up room, and the water molecules, there wouldn't be enough room for as many water molecules. So that means then the water would have a lower concentration on the right and the left. And so the water would move out of the cell. So if you put the cell into a very strong sugary solution, water would move out by osmosis. Okay. Right, so let's have a think about some practical effects of that then. So what I've got here is I've got 3 plant cells, and this one here is normal, okay? That's got the normal amount of water in it. This one here is described as being turgid. You can see it's sort of bulging, and that's because this cell has been placed in pure water, or a very dilute solution. So that means the concentration of the water outside the cell is higher than it is inside the cell because of all the things dissolved in here. So water is going to move into the cell, and it's going to make it sort of bulge. Now the cell won't burst because plant cells have a cell wall, a cellulose cell wall, and that will prevent the cell getting so big that it actually bursts. Okay. Let's have a look at the cell on the right here. And we describe this cell as being flaccid, and that cell must have been placed in a solution of a high concentration, maybe of salt, maybe of sugar, maybe of something else. Let's say sugar. And what will happen then is the concentration of water is actually higher inside the cell than it is outside the cell. So the water is going to move out of the cell, has moved out of the cell to effectively dilute the sugar solution that it's been placed in. and that means it's got this shriveled looking appearance. Okay, right, now this is quite important if you think about you can actually show this in an experiment. So I'll just 1 minute. We'll just move those plants out of the way in a minute. So you've got 3 beakers there, and this is a piece of potato, a chip, and let's say that this one is pure water. This is a dilute solution of, let's say, dilute salt solution, and this is a very strong salt solution. Right, so let's think about the pure water. Well, the concentration of water outside is going to be greater than it is inside the potato, because you've got loads of, the potato is made up of loads of potato cells, and so there's lots of things dissolved in those cells. It's starch, grains, proteins, and all kinds of things. So water is going to enter the potato cells by osmosis, and so the potato chip should get longer. So you can leave it and you can measure it, and there's a required practical, which is described later, and the potato chip will get longer. Now, if you put the potato chip in a very strong salt solution over here, effectively the concentration of the water is higher inside the potato cells than it is outside, because there's so much salt in this solution here. The concentration of the water is low. So that means water will move down the concentration gradient and leave the cell by osmosis across the semi-permeable membrane. And so that will, the potato chip will get shorter. It will shrivel up. You can actually measure it and measure the length of it in millimeters, and it will actually get shorter. Now, if this chip has been placed in the concentration of a salt solution, which is more or less exactly the same as the concentration of solution inside the cell, there'll be no net movement of water, because you've effectively got the same concentration of water inside the cell and out, so that there's no reason why the water should move. Okay, so this has fairly important practical effects. So if you look at, if you've got a house plant and you don't, and it's watered, it's going to look like that, and that's because water will enter the cells by osmosis from the soil, and it will make the plant turgid and rigid. Now, if you forget to water it, what happens? We say this plant has wilted, because the cells, there's not enough water in the pot to enter the cells by osmosis, and so the cells all become shriveled, and the plant doesn't have enough rigidity to support itself. So the leaves can't stick out and they can't get any sunlight. So, and if you don't water that plant and make it turgid again by making the cells take in water by osmosis, the plant is going to die. Right, let's see what happens with, so much for plant cells. Let's look at animal cells. So the easiest thing to look at here are red blood cells. Okay, so if you get some red blood cells, if you put the red blood cell into pure water, and this one, the cells burst. Okay, so all you're left is with just membranes. And why will the cells burst is because, well, inside the cell, you've got a high concentration of stuff dissolved in there. You've got hemoglobin and your other things dissolved in there. And so if you put it into pure water, the water is going to enter the red blood cell. There's no cell wall in a plant cell, in an animal cell, to stop the cell bursting. So the cell will just swell up until eventually it pops open and it will release all the hemoglobin into the solution. If you put if you put it into a very, if you put some red blood cells into a very strong salt solution, so you've got so much salt dissolved in the water outside that effectively the concentration of the water is lower outside, all the molecules, water molecules, will leave, and it will lose its typical biconcave shape, and it become sort of shriveled up like this. Okay. So is that important? Well, it is important, I suppose. If you are given, for example, antibiotics, if you by an intravenous drip, if you've got really bad infection and they have to put the antibiotics directly into your veins rather than just taking tablets, then you've got to make sure that the solution of the antibiotic, you can't dissolve it in pure water, because if you do, you're going to end up bursting all the red blood cells. So it has to be dissolved in what is called isotonic salt solution. So what that means is the concentration of the salt solution you dissolve it in is more or less exactly the same as that inside the cell. So that means the water is not going to leave the cell by osmosis, and it's not going to enter the cell by osmosis. Okay, so just to recap, and what do you need for osmosis? You need a semi-permeable membrane. Okay. Semi-permeable membranes, all cell membranes are essentially semi-permeable membranes.